My name is Timo Dickscheid. I'm working in Jülich, and I'm part of the HIBALL consortium and of the BigBrain community, and I'm also working in the Human Brain project in Europe, where I help to build up an atlas of the human brain. And today I will talk a bit about how BigBrain is integrated into this atlas, and also how that atlas is structured, and how it is embedded in the EBRAINS infrastructure that is being built by the Human Brain Project. The idea of my talk is to first give you a conceptual overview, and then we go live into some of the tools. And at some places, you will have the opportunity to try things out yourself just in your web browser. That is not necessarily needed. I will show it as well, but if you have a recent version of Firefox or Chrome open up and running, you will be able to try out a few things while I'm talking. I will let you know at that moment, and we will also paste links and URLs into the chat when I'm there. So let's get going. Before we talk about a brain atlas, I usually like to discuss a little bit the idea of an atlas at all. So when we talk about brain atlases, we typically mean a few different things. We mean that there is a reference space that gives us a coordinate system, like you can see here, the coordinate system of the Earth, and one of the brain, which is the very famous MNI reference space. And then there is a map that typically tells us where we have different regions in that coordinate space. We see a map here of the Earth with the countries, and we see a map here of the cytoarchitectural parcellation from the Jülich brain atlas in the brain. And lastly, there is typically a taxonomy that tells us what the names of these different regions are, and possibly also what the relationships between these regions are, and some properties of those regions. And when you deal with atlases, I think that most of you are neuroscientists. Some of you might already be working a lot with atlases. I think you will typically have in mind that you have something like this. Yeah, you have a reference image, a reference volume or reference surface, and you have the coloring of the surface. And for sure, you have some kind of text file or structured file that tells you what the colored regions are. So I think most of you will typically expect something like this to work with an atlas. The idea is now for the multi-level atlas for the human brain that we are building in the Human Brain Project and in eBrains, that we go a bit further than that. And that is very similar to what you know from the Earth. If you have a book atlas of the Earth, you typically find more than one map. You might also find maps of the topography or climate in the Earth on different pages that you could compare with each other. And that is very much the idea that we also follow here when we say that we want to build a multi-level atlas of the human brain. And in this talk today, I will try to explain this concept of a multi-level atlas, the role that BigBrain plays in the multi-level atlas. Then I will show you how this is embedded in the eBrains infrastructure. And we will then use this atlas a little bit with the tools that are available. And as I said, there might be a chance for you to put your hands in the browser and play with it a little bit. We will see how we work with parcellations and different spaces, how we get access to data features, and also how we could run some analysis using those atlases. And at the end, there will also be a small outlook of the features that will be coming up. So in particular here, at this point and at this point, I will make sure that you are able in your browser to also try things out while I'm doing it on the screen. So the concept of a multi-level atlas. Here we see a screenshot that comes from the viewer in the eBrains atlas services that shows the MNI space. It might be very common for you. And this here are some maps. In particular, these are the cytoarchitectonic maps from the Jülich brain probabilistic atlas. And this here is a complementary map. It shows white matter fiber bundles that have been computed from a neuroimaging cohort at Neurospin in Paris. So this is very complementary. This is a map of short fiber bundles, also coming from the lab of Jean-François Magin, Neurospin, which is again complementary to the last one. And here we see again a different thing. This is an atlas of functional modes, also extracted from neuroimaging, from many fMRI scans from different individuals by the lab of Bertrand Thirion. You probably know that parcellation, which is available at different granularities. So here you see a higher granularity, a finer granularity of this same atlas. So in principle, by providing different complementary maps in the same space, we already have a certain way of providing multiple levels similar to the atlas of the Earth that are shown in the beginning. And now comes BigBrain. You have already seen and heard a lot of BigBrain. I think I do not have to explain too much again today. Much of this school is centered around BigBrain. You have even got your hands on some data sets that include BigBrain. You have seen that it's big. It's not easy to retrieve the whole thing quickly to your local disk. And in the multi-level atlas in EBRAINS, we support BigBrain as an additional template space together with the M&I spaces. And as you have seen, BigBrain has a much higher resolution of 20 micrometer at the full resolution, isotropic in 3D space, but we could still provide it to the same tools and put links between them. And this means we have links between different spaces. We also have maps, cytoarchitectonic maps defined in the BigBrain space, which are comparable to those that I've just shown you in the M&I space at the millimeter scale. Just very quickly, the BigBrain has been built from more than 7,000 histological sections, as you can see here on the right. And I think you have learned this already today. It is a 1 terabyte data set that we reconstructed in 3D in many years of work, has been done in Montreal at McGill, and the data has been prepared in Jülich, and we worked together over many years to produce this model, which is now freely available, and you can find it at different places. One of them is this EBRAINS atlas with the viewer you see on the left that we will soon see. There are other ways to find it. You have seen data storage, where you can retrieve it. You have seen how it is integrated also in the Canadian tools that we've seen in the last sessions. Now in the multi-level atlas, the idea is really to connect these different template spaces, at the moment the M&I spaces and the BigBrain space, across the scales. And one way to do this is to have spatial transformations between the coordinate systems. In particular, we have nonlinear spatial transformations. In the EBRAIN system, they come from these publications. So they are based on a nonlinear warping that took into account Salkel constraints. And these spatial transformations, they allow us, given a coordinate in the BigBrain, to say where approximately the same coordinate would be in one of the M&I templates. So this gives us a coordinate mapping from one space to the next across the scales. This is one way of connecting these different template spaces, but there are more ways. I already indicated it a few slides ago. Another way is if you have comparable region definitions, which we can see here. So we have a number of cytoarchitectonic maps in the BigBrain, which are mapped at the full microscopic resolution in 3D. Yeah, some of the maps you see here, they are really mapped at great detail in thousands of histological sections very precisely. I will talk about this in a minute. And so we have a relationship here between corresponding regions that have been mapped in many individuals into the probabilistic maps you see here in the Collins space in this example. And we have also mapped, according to the same principles, these areas in 3D in the BigBrain in this single subject space at full resolutions. So this also gives us a link across the scales. And just to talk a little bit about how this is being done, the cytoarchitectonic mapping looks at the distributions, the patterns that the neuronal cells are building. You see here 2 patches from microscopic scans from human brain in areas V1 and V2, and you can see here how these the neuronal cells build cortical layers with very different properties. In these two cases, it's quite obvious where how these differences are. You can see here in this cortical layer, we see quite large neuronal cell bodies. That's also the case here, but not as many. And you can see here that the density of cells in this layer is much lower, and there are not so many large cells. And these properties can be detected, and that is what for the Jülich Brain Atlas is being done for many, many years and collected in many, many probabilistic maps by analyzing cortical profiles, as you can see here, and looking at how the gray values in these histological sections behave along the profile, and running a statistical analysis that basically confirms that there is a point, which you see now here, where the pattern of this cytoarchitect of these cortical layers changes significantly, and tells us that there is a different structural area starting at this point. So there is a way, and this is procedure by Schleicher et al. To confirm in an observer-independent way that at this position there is a border between two areas. This is being done for many years in Jülich, and what we get from this are sections with maps, histological sections with maps of cytoarchitectonic areas, and we can aggregate those into 3D in this particular postmortem brain. We typically do this at the millimeter resolution. So while we have the map in the individual section at the micrometer resolution, we typically do not have these maps in each of the sections. In many cases, we don't even have all the sections of the brain. And so when we put them together into 3D, we cannot provide the full resolution as we have it for the big brain in any case, but we can typically provide such a 1 millimeter map, as you see here on the right. Now there is a very important aspect of the human brain, that is the remarkable variability that we have. You see here these 3D maps of areas V1 and V2 in 10 different postmortem brains as they have been used for the probabilistic atlas, and you can see how tremendously different they are, how huge the variability is. So it doesn't help us just to have one of those maps, and that is why the Jülich brain probabilistic maps are aggregated across many postmortem brains, as you see here, typically 10 postmortem brains. And the way we provide them in the atlas is by registering the individual 3D maps into the MNI reference space and building a probability map in the reference space, as you can see here on the right. And taking all these different probability maps for the different regions, we can then build a simplified maximum probability map that gives us a coloring of each voxel, of each surface point by the region that has the highest probability. But please remember, if you have such a probabilistic atlas, this here is a simplified. This is actually a simplified map. And it's always good when you see the map to remember that you have these probabilities in the background, which are much more meaningful because they really give you some capturing of the variability of the human brain and access to really a lot of work, many, many years of work of neuroanatomists of mapping these areas in the different brains. Now, when I go back, I have told you that we have in BigBrain several cytoarchitectonic areas where we have a full 3D map. And this means there we took each of the sections where you see this area of this BigBrain sample and mapped the region in each of these areas. And for area V1, this is much more than 1,000 sections, so it would not be feasible for neuroanatomists, even with the tools that we typically use to do that by hand. And it would also not work nicely by interpolation. So we are looking for support from machine learning, artificial intelligence in doing that. And since some years, and this is also an important aspect of the HIBALL lab that is running the school, we try to use deep learning methods to perform these brain mapping tasks. And this has started several years ago. Hanna Spitzer was the first PhD student in our lab who worked on that. We basically took such annotations in histological sections from an expert and tried to teach a deep learning algorithm to predict, to do a segmentation of the image that also finds these areas. So to look at each pixel and try to color the areas in the same way. And over the years, we have made good progress with this. So we have now deep learning networks that are quite good in mapping some of these areas. And we are working on networks that would be able to map almost all of these areas. I will show you some more results of this. But first, to understand the problem a bit better, what these machine learning algorithms do is they see such a cortical patch, as you see here, 12 of them, and they have to tell which brain region is it, which cytoarchitectonic brain region is it. And for the neuroanatomists among you, there are some which are maybe easy. So you might easily be able to tell what this is. I'm not, I know it, or what this is. That's okay. Yeah. But then you have also places, if you want to map in each of the histological sections, you have places where you have a strong curvature, like here. Or you have places where you have very oblique cutting angles, like here, where it's really not easy to see. And then you have also sections where you have histological artifacts. Even if you have a very professional lab and experts in the lab that do it very carefully, it's unavoidable that you have histological artifacts from time to time. And if you want to map each of the sections, the algorithm has to deal with all these problems. And this is very difficult. However, we are now at the point where we have some of these areas mapped in full 3D in the BigBrain. Here are two, V1 and V2, and the recent preprint that you can find on the web from Christian Schiffer. And this is what you have seen a few slides ago in the BigBrain, and which gives us a nice link across the scales. So we have a corresponding definition of the region at the micrometer resolution in 3D with the probability map that we have at the millimeter resolution. Now, there is another aspect of a multi-level atlas, as we understand it, and that is to link information from neuroscientific studies from different modalities to a brain region. This is something that we do in the Human Brain Project. We collect studies of different brain regions, or histological studies, in vivo studies, and we perform curation to carefully assign the results of these studies to brain regions. Here you see just some examples. Yeah, histological studies. This here is an image from a polarized light imaging, which shows the fiber orientations in a section. These are neurotransmitter receptor distributions. Here you can see individual cell bodies, fluorescence microscopy, and here on the left you see a whole brain connectivity network. Such examples can be linked to brain regions from the atlas, and they provide you multimodal characterization of the regions in the atlas. And this is the third level of this multi-level atlas that we integrate and that you can access to understand the organization of the brain. So we have mostly three links that provide this multi-level aspect of the atlas: different spatial resolutions, complementary maps that have been that describe different principles of brain organization, and multimodal features of different brain regions that describe the different facets of that particular brain region. Now, how do we embed this in infrastructure? One important building stone of this is in EBRAINS, the data curation services. So the Human Brain Project provides real humans, really teams of humans that help you to curate and share your data in the EBRAIN system. So they really assist in bringing the information for a particular data set into structured metadata schemes, rich metadata schemes, describe it in a coherent way, and assign brain region information and so on. This will then go into what we call the EBRAINS knowledge graph, which is basically a metadata database that has a search interface, but it also has an API where you can search for the metadata, and the actual files, they travel to a cloud accessible storage system. And the EBRAINS Atlas services connect to this knowledge graph and storage systems, and they provide ways of interacting with brain regions and with the multi-level atlas that I've shown you. But all the information of this atlas, the templates, the brain region maps, the multimodal features, these data sets, it's all stored in a knowledge graph, curated data with deep information stored in a knowledge graph that is accessed through these atlases. And another aspect is that these services also connect to high-performance computing systems, which allow us to compute these maps, which I've shown you, and also in the future, in a similar way as you have seen it now for the BigBrain system, to use these atlases to run analyses on the high-performance computing systems. But today I will talk about these atlas services, and in particular we will have a look at two different things. At the 3D atlas viewer, and this is also where you will have a chance to get your own hands on it. And I will in parallel also show you a Python client that can do much the same ways that we will do in the atlas viewer. This Python client, however, is still at the alpha stage, so it is not stable and it does not yet have a version 1 release, but it is publicly accessible and is already working. So we will see a few examples from this Python client. We have a quick check of the clock. Okay. So let's dive in a bit. That was for the conceptual part. Now we go a bit away from the slides and have a look at the actual systems. So we will go now to the interactive atlas viewer and try out a few things. Before we do so, and I think Susanne will also post a link, but just to let you know that it is not difficult to find this. So you can just go to ebrains.eu. I hope you see that here on my screen. And there, if you just scroll down, you already see atlases. Yeah, you can click on atlases. And there, if you scroll down, you already find the Human Brain Atlas and some tools. But if you click on the Human Brain Atlas, you find here Explorer in the interactive atlas viewer. And here we go. We see a Human Brain Atlas in the atlas viewer. And that is also where you get when you click the link that Susanne provided you there. So let me show you a few things. If you like, please open your browser and launch this viewer. If you have a recent version of Firefox and Chrome, it will work. If you run Safari with the current version, it will not work because it requires WebGL2 support. Good news is that the technology preview of Safari works. So in the future, Safari will also work. But today, Safari is not working. Firefox and Chrome, a recent version, should work if you don't have a very exotic computer. Let's not do a lot of things. Let's just start by a few simple things. I talked until now about complementary parcellations and different template spaces. And you find them here on the left. I hope you can see my mouse. On the bottom left, there is a button, select layer. And this, a little bit like it's done in Google Earth, it gives you, it shows you different layers that you can access. We are now in the ICBM 152 space. And here we have access to these fiber bundle maps. So you can just click on the fiber tracks. You see long bundles, short bundles. There's a new version of short bundles computed from the HCP dataset. And if you wonder what this actually is and where it's coming from, just hit the little eye icon. And you see a description of this dataset. You also see a publication. So you know, this is from the French people. I told you it's from Jean-François Magin, and you can read it here. And you see here a button, Explore. And if you hit this Explore button, it takes you right away to the knowledge graph. I will do that now here. But you can also just stay in the viewer. And you see, this is the data set that is associated with this map in the eBrains knowledge graph, where you have the full metadata description of this atlas. And you could also download this map here as a file. You see the related publication. You even see that it has been computed from 78 different subjects, female. There's rich information here about this one particular parcellation. You also have citation policies and so on and so on. I close this again, go back to the viewer. What you also see is that you can download this map here. You can download a zip file of this map, which you would also find in the knowledge graph, and you can pin this data set. I will come to this later on. So if we select it, the map changes. That's no magic. And you also find here the functional modes, the Diffumo Atlas from Bertrand-Thirion. Again, if you hit the eye icon, you see here the information, and it can bring you to the knowledge graph. And what you also see here are different template spaces. There's the Collins space, and there's the BigBrain. So if we select the BigBrain, you will actually see what you have already seen in the early and excellent anatomy tutorial by Nicola this morning. And on my machine, it might take a little moment to load the surface meshes of the BigBrain, since I have a few children using their computers in parallel, and I'm running this video conference. But here you see already these cytoarchitectonic maps that I've shown you. And you can switch also for the BigBrain between different maps. We can open here also these beautiful maps of cortical layers from Konrad Wachsdühl, where you will learn a lot about those tomorrow and learn it right from the expert. Just quickly, we talked about pinning data sets. So if I just do that again, I hit here this information icon and I say pin this data set, you will actually see a number flashing up here on the right. This is actually kind of a shopping cart for data sets. We will come to this later on, but it will just remember that you were interested in this maximum probability map, and it just pins it here without downloading it. So far for selecting regions. I quickly check the chat if there are any problems or questions. I do not see any here. Then let's go a bit further. What is the time? We still have time left. Let's have a look at the Python client. You will not be able to replicate this now live. I just show you that you can do the very same thing in a Python client, and the Python client is called Brainscapes. So we are now in a Python notebook, and I can just import this Brainscapes module. I import this as BS, Brainscapes, and then I can do just the same thing. Yeah. So I can say, give me the multi-level human atlas, just in a way that in the browser, sorry for this. In the browser, I can also switch here between the human atlas and the atlas of the rat and the mouse brain. In this very same way, I can here select the human atlas, and I can select the parcellation. The parcellations are clearly defined. I select here the probabilistic cytoarchitectonic maps, just as we did it in the viewer. And I can select the space. The spaces are also clearly defined in the library. I select the ICBM space, just in the same way as we have done it in the viewer. And then I can retrieve the maps. That is exactly what I have done in the viewer. I run this cell now, and I use the NeLearn library to visualize it. And you see here the same maps, just left and right hemisphere, plotted in Python. And I can also access here from the parcellation a description, which shows you just the same text that you have seen in the viewer, pointing back to the knowledge graph. And I can, of course, do that as well for some of these complementary maps, dictionary functional modes, long white matter bundles, superficial white matter bundles. If I run this, it will retrieve the maps from their storage locations online. This might take a moment because I'm now querying three different maps, so it might take a moment. But here you see I get exactly these complementary maps. And then we go for selecting brain regions, but I will show you in the viewer again what it means to select brain regions. In the viewer, selecting a brain region is a very simple thing. You just double click the region. Yeah, so let me go for here for our best friend area V1. And when I double click it, you might now do that yourself. When I double click it, it will load the probability map that I talked about. So once I select a single area, it shows the probability map that it retrieves from the background, and it opens a side panel with information about that brain region. I will come more to this side panel. Just before we go on, I close it again, and you see the selection is visible here on the bottom. I hope I'm not going too fast for you. I will check in the chat quickly if there's I just see a question that I answer right away. So this is a tool for working with the existing atlas. It is not a tool for annotating or editing the atlas. There will be some annotation support, some annotation tools linked to it, but at the moment we don't have any. So this is a, let's say, read-only tool with respect to the parcellations. So when I click here the X on the bottom, it deselects again. And I just show you that you have access to the full region hierarchy here on the top. So I can also search for V1 and select it from here with the exact same effect. Yeah. But what is more important is, instead of searching it, here's this little tree icon. I also see the full region hierarchy, and I can browse the full region hierarchy here. I can, for example, put in IPL. And it shows me the different areas here of the inferior parietal lobule. So you might want to try that out a little bit for yourself. We don't have terribly much time, but I talk slowly, so just play around with it and post questions if needed. We basically have seen now two of these levels. We have seen different scales and we have seen complementary maps, but I also talked about these multimodal data features. And we can explore these multimodal data features also in this panel. So once we select a brain region, in this case here V1, in the side panel I see a description. This is the description which again comes from the knowledge graph. So this particular probability map is also a data set, a curated data set in the knowledge graph. And I just have here a copy of the information text. Yeah, but I can here on the top, where is it? On the top, I can, probably I was a bit fast. You see this little icon for the additional information here on the top. And it again brings me to the data set card for this particular probabilistic map of HOC1. And you see here the rich description. You have backlinks into the viewer. So it goes not only from the viewer to the knowledge graph, also the knowledge graph has links into the viewer. And you find here the individual files. If you want to download them, you find the related publications. You see the subjects here, the postmodern subjects that have been used to map this probabilistic region. Yeah. I told you that they come from 10 postmodern brains typically, and you see here some information about these 10 postmodern brains that have been used for this one particular map. Going back to the viewer. Explore another template. This shows you that the map for V1 is also accessible in the BigBrain space. So if I select BigBrain here, it will open the BigBrain and show me the map of V1 in the BigBrain. And the map is also available in the Collins space. I can go there from here. I'm not doing that right now. I was talking about the multimodal features. And if you select regional features here on the left, you see that there are some data sets linked to this region. And I will show you some of them now. The first one for me, and I think that should be the same for you, is a link to the human intracranial EEG database in the Human Brain Project. You can click on it and you see a little exclamation mark here, which tells you that this data set is currently reviewed by the Data Protection Office regarding GDPR compliance. I can tell you that this will soon disappear, but for now, we can see some information, but we cannot access the actual recordings because of the you would need to authenticate and sign some agreements. But what we can see here is a description of this study, and we can see the electrodes that overlap with the selected region V1. So you see here on the left 2 electrodes that go into V1. And you can see here the cognitive tasks that were associated to this. And you see the 15 different contact points. And I can click them and the viewer would go there. Same for the second electrode. The data set has more electrodes. Yeah, these are just the 2 electrodes that we found overlap with the map in MNI space. Okay. This is still slightly disappointing because you don't see the actual EEG recording, as I said, but it will be accessible soon. And if you go to the knowledge graph again here, to this Explore link, you will actually find more information, and that is where you will already very soon be able to see the data. The Atlas viewer will be a bit delayed, but you can also expect until the summer, I would guess, that you find the actual recordings here. I'm going back to the side panel for area HOC1. I have to speed up a little bit, but I fetched the second data set here. These are density measurements of receptors. And you can see here, they come from Silis and Nicola Palomero Galafa. So you see here measurements of densities of transmitter receptors, different transmitter receptors, and you can play with them. So you can select NMDA, and you see a cortical profile of this density that has been measured. You can also find here an example image of the original autoradiograph from this area. You can download all of this here, as you can see. And you can also pin the data set, by the way, and find it here in your shopping cart. So there is already a lot of data linked to brain regions, not for every brain region. It's not fully complete, but this is growing. Lastly, you find here a panel for connectivity with a list of different connectivity matrices that have been curated into the system. And since we selected area V1, we see here the connectivity profile from this selected connectivity matrix from the 1000 Brain study. We see here the connectivity profile going out from the selected area HOC1, and we see the connection strength of other areas. And we see also the brain here colored now by the connection strength. And we can go through these different areas and actually also select them and go there. So in a way, we can here travel along the major connections. We can even quickly select here the other area that has a strong connection and see the probability map. So you can interact with the connectivity matrix, travel a bit through the brain. This is all visually. You explore the things. Yeah? This is not, in the viewer here, it's not in particular meant for data analysis or anything, it's for exploring it. But you can download all of the data and work with it. Just checking again. This was also a question, are these Atlas data downloadable? Yes, they are. Yeah? And all these data sets are usually in the formats that you would expect. So you can download the map of the Atlas if you look at the information item from the Atlas, and this, if you download it, this will be a Nifty file. But the receptor data has different formats for sure. This is a cortical profile, so this will be a TSV file typically. But yes, you can download almost everything with the exception of the data sets that are still under the GDPR review that I just talked about. Let me show you how that would work in a Python client in a programmatic way. So in a Python client, you can also, you can also select brain regions. Yeah, you can select the frontal pole and you get the whole subtree of the sub areas of frontal pole, but you can also select V1 left and it will select this particular area. You can get some properties of these brain regions. So you can retrieve the probability map by selecting the specific map. And then you have to provide a space because this map is available in different spaces. And you can plot the probability map of FP2 in that case. You can also access some properties like the centroid and the surface, the surface area, things like that. And what you can also do is to retrieve these data features. Just as in the same way we just saw it in the viewer, there's a method query data on the atlas object, which takes into account the selections you made. So if I select region V1 and then query the data for a particular modality, there are several predefined modalities. One of them is gene expressions, and I can put in a particular gene. If I do that, it will extract features in this area for me, in that case, gene expressions, which are taken from the EBRAIN atlas. I have run this notebook a few minutes ago. Otherwise, there would be a little information that it's now accessing the EBRAIN atlas and informing you about it. And you can see here the probe locations from the EBRAIN atlas that have been retrieved for this particular area. In the viewer, I have shown you how to access receptor distributions. This works very much the same way. Again, I would say query data, but I choose a different modality, receptor distribution, and I get these features, and they are already prepared in Python that you can easily plot them. You know, they already provide you a nice plotting routine that you can see the fingerprint and the cortical profile. Be aware, this is alpha, so there might be errors, and this API will most probably change over time. I see in the questions that you ask if this notebook is openly available. Yes, it is. I will share it after this session. And there's also a public repository with the Python code that has example notebooks right in the source code. I will share links after the session with you. Lastly, we looked at connectivity matrices. And again, this is the same thing. I call the same method, query data, with another modality, connectivity matrix. Since this is a matrix, a full matrix, the query data routine knows that it shouldn't worry about the selected region. It just worries about the selected parcellation. So it gives us connectivity matrices that match the selected parcellation, which was the Jülich brain atlas in this notebook. And we see here 4 different connectivity matrices, the one from the 1000 brain study that you also saw in the viewer, and some more that have been computed by Simon Eickhoff's group. And the one from the 1000 brain study is coming from Svenja Kasper's group. And these data features, they are growing. Yeah? So the curation teams in eBrains, they are working, and this is all growing. Just to inform the organizers, I have the impression I will eat up most of the time, but I'm also answering questions while I'm talking, so I hope that is fine. There will not be 15 minutes left for questions, but I hope it's fine to answer questions right away here while I'm talking. So we have now seen how we could browse regional data features with these tools. And as a last example, I want to show you how you also can perform some basic analysis using these tools. And this is something we are doing now in the next years a bit more. In the past, we have not concentrated so much of analysis, but there's one tool that I want to show you today, which is available already. It's the JUGEX tool that has been proposed by Sebastian Pludau, whom you will meet tomorrow, and others, 2018. And it was originally implemented in MATLAB. The MATLAB implementation is for sure still valid and available. You find the link here. But we have also re-implemented this JUGEX tool because it uses the cytoarchitectonic atlas. We have implemented it in 2 different, actually we have 1 implementation, but it is accessible in 2 different ways. One way is a plugin of the Atlas viewer, so you can use it interactively in the Atlas viewer, and I will show that now. And the other is using this Brainscapes Python client in Python. Yeah, so there are 2 ways that you can use it with the tools I've just presented, but for sure you also find the original MATLAB implementation, and you get almost the same results. We are still checking that we might hopefully get same results up to numerics, but this is always difficult. So let's also go into this. I think we might have the time that you try this for yourself. So you can just stay in the viewer, but I think Susanne did probably send another link. So JUGEX is also documented on the eBrains web page. If you go back to eBrains and select atlases again, there's a description for JUGEX here on the bottom. You see it here, JUGEX, which describes it. And you find here a link to the Atlas viewer where you can use it. you find here a link to a Python notebook. That is actually where you would also find the other Python notebook examples. You find a link to the MATLAB implementation. And we will go now to the viewer again. Let me just copy in this link here into the chat. The thing is that JUGEX currently requires you to select the previous version of the cytoarchitectonic maps. You can do that by clicking here in the left bottom, where you find different versions. But if you follow the link that I just pasted in the chat, you will already have the right version pre-selected. Yeah. So ideally, just if you want to try it out yourself, click the link in the chat. You see again the cytoarchitectonic atlas and MNI space here. And if you go to the top right, you see here a little icon that gives you access to plugins. And there you see JUGEX as a plugin. If you click on it, you get a toolbox, a little graphical user interface, where you can perform a JUGEX differential gene expression analysis. What it does does in principle an ANOVA analysis of gene expressions in 2 different regions of interest. And you can select regions here. If you click on this little radar for the first region, I will select area V1 here in my case. And you can select the little radar for the second region by clicking here. And just click the region you're interested in. I select area V2. Yeah, so we have now 2 regions configured. You could add more areas to each region if you like, but I just select V1 and V2. And then you have to select candidate genes. Yeah. Some of you might know gene names by heart. I don't. I just learned these here by heart. TAC1. And I will also add another one, which is MAOA. Don't ask me what these are. I'm a computer scientist. And now we have the analysis in principle configured. Yeah, we have a region selected, first region selected, area V1, a second region selected, area V2, and we have selected a few candidate genes, in my case, TAC1, MAOA. There are some settings here. I'm not talking about all the details. I will reduce the number of permutations a little bit on my side. You should use many permutations for the ANOVA. I use only a few for this demonstration, so it's going a bit more quickly. And then I can run the analysis. I click run, and this will initiate a computation on a back end, which has not been proof tested for many people using it in parallel. So this might today be interested. If you are all doing that now, I would be interested if the server loves that or not. At least for me, it worked. So the analysis passed. I can see that here. I see now here the p-values coming out of this analysis, which are not the right ones, but I just used so few iterations only. And you see here the locations of the probes that it has been extracted from the Allen gene expression database for the different genes. And if I enable this little button here, it will also show me in the 3D view here, which I can also maximize. Did you see that on the very bottom right? There's this little square that you can hit, and it will maximize the view. So you can see here now where these locations are that we used, and you see that it has used them on the left hemisphere, and they are here in the occipital lobe in areas V1, V2. And for sure, you can download the resulting p-values and probe locations. You will get a TSV file if you download it. Just try that out. We have 10 minutes left. I have not so much left, so I will maybe wait a minute. This was probably a bit quick. And then I will show you that this can also be performed in Python. And the good thing is, what you have done here in the viewer uses the Python code anyway. It is not a separate implementation. It uses the Python code. And so one nice thing is you can just get the Python code of what you have done here by clicking here on the Jupyter Notebook link. There are still some flaws, but if you click here on the Jupyter Notebook link, which appears when you have run the analysis or configured, it actually opens a documented notebook that does the same thing using the Brainscapes client. Yeah? So you get here a notebook that does the exact same analysis. It's exactly the code that was executed in the viewer in the background on the server that you can find here. You cannot yet execute this notebook right away here, but you can download it and execute it on your own machine. But that requires you to set up the Brainscapes client. And I didn't cover this today. It would have been too much to cover how you install the Brainscapes client. But I'm happy to send a few links and instructions how that could be done. As I said, we are not yet at the version 1, and you can already pip install Brainscapes, but you will not get the most recent version which supports this analysis here. So please stay tuned and please be in touch with me if this is of interest. I'm really happy to help you out if this is interesting for you. I just opened this same notebook in my Jupyter notebook here as a real one, and we will quickly go through it. You need an HPP authorization token if you use the Brainscapes client. This is something that is documented here in the notebooks and also in the Brainscapes client. HPP gives you access to these data sets for free, but you need to authenticate. You need at least for many of them. And for using it here in a programmatic fashion, you really need such a key. And it is in the documentation how you get that key. It's quite easy. So again, I instantiate here the Brainscapes library. I select the human brain atlas just as we have done before. I select the map. Here I enable a specific setting that you wouldn't necessarily need to do, but I tell Brainscapes here, instead of using the static maps, the maximum probability map, it should prefer to threshold the continuous map, the probability map. Is enabled here because the original MATLAB implementation is doing that as well. You would not necessarily have to do that. Then I just define these two candidate regions. These are, you will see it's the same ones that we used in the viewer, V1 right, V2 right, and the same two candidate genes. And then running the analysis is as easy as this. I generate this analysis object, differential gene expression. It gets the atlas as an input, because the atlas tells it all about the selected region and the spaces and what not. Then I let it know about the candidate genes and about these two regions. And you can see here it will use the probability map. It retrieves the probe IDs for the different genes from the Allen atlas. And then I can run the analysis. I will also choose here a few less permutations. Run the analysis. And you get a result. Yeah. If you use a higher number of permutations, you should absolutely get very similar results. If you put in a random seed, you should get the identical results. So here you have also the possibility to put in a random seed to verify that the computation is doing the same thing. And then we can also here in a very easy way retrieve from this analysis the samples again with the information, and for sure also display the locations that have been used from the gene expressions in MNI space. So you can see here the probability maps and the locations that had been used. In that case here on top, I plotted area V1, and on the bottom I plotted area V2. So if you're interested in this Python client, which is at the early stage of development, please get in touch with me. Yeah? I'm happy to connect you, to provide you links and access and support. But please please be warned that this is early development. There might still be errors, and we might change the API of this. All right. Are there questions? I'm checking the chat here. I do not see any new questions.
I did mention Brain Browser, Timo, you might want to mention.
Oh, yes, yes, I see it here. Let me just click it for everybody. So if you want to explore in particular surface maps, that is something that the tools I have shown you are very bad at. More precisely, currently, we are not really supporting surface maps. But
if you go to the macaque data set, that will probably be a nice example, and spin the brain.
Exactly. Yeah, so this is the page here for the brain browser tool, which you can execute right in your browser. And here's the surface viewer. You see. And you find the link in the chat. So I think we are doing pretty well in terms of time. This is almost the last slide. So where are we going here in particular for the EBRAINS Atlas part? I'm not talking about the future directions of HIBALL. I'm talking here about the EBRAINS Atlas part. We will add surface support for the free surfer average to the tools that you have seen. This will come this year. We will produce a stable release of the Python client, and it will also come with an HTTP REST API. The version 1.0, I also hope that we will have it this year, but I don't want to give a fixed date here. But there will be a stable release coming in the nearer future. And I already mentioned that on the analysis side, there will be more. So we are working now in the remainder of the Human Brain project on toolboxes for analyzing. There will be functionality of the SPM anatomy toolbox included in the eBrain systems that will be developed together with Simon Eickhoff, who is the author of the anatomy toolbox. There will be a connectivity toolbox to work with connectivity data and to connect to the NeLearn library. Here, Bertrand Thirion is contributing and cooperating with us. We are also planning to provide support that you can project individual MRIs into these tools to overlay, to easily overlay an individual subject scan with the Atlas data. And one thing that I'm very interested in is that you will get big data access through these brain regions. So what you've seen now basically doesn't give you access to the to really big data. Yeah. So I shown you that you could download the maps in the MNI space as a nifty file from the viewer and also from the Python client. If you would want to download the BigBrain and the maps of the same areas in the BigBrain, it would not be possible because that is terabytes of data, and you don't want to download these from the browser. So we are working on that. There will be ways of very easily accessing the large data as well in a coherent way. We are working on that, and also to retrieve distributions of gray values of cell densities. But that is not coming this summer. This is coming in the, well, rather across the next 2 years, but it will come. So there will be big data access through these same tools. And this will also use data-led and things that you have seen earlier. And my last slide is the most important next thing. We want to be in touch with you. Yeah, much of what you have seen might really have a bias from the people in the Human Brain project, from the people developing it, as always. But we really want the input from the community, and this is now, particularly this year, very important for us. So I'm really, really happy to receive feedback, criticism, requests, whatever. You can get in touch with the eBrain support. You can be in touch with us through HIBALL, through the forum for BigBrain, in particular if it relates to BigBrain things. But please don't be afraid just to write me and myself an email. Just get in touch with us. We are more than happy to hear from you. Yeah? And one last thing, here's my thank you slide. One last thing that I don't have on the slides, but that I would like to add. No, I will do a quick wrap up at the end today. I have a few minutes. I will add this there, actually. I will have some time to prepare it. So I'm ending here, and thanks a lot to many people who contributed to all of this. This is not my work in particular. There are many collaborators from the Human Brain Project, from the HIBALL project. Many of those people are here. From my working group in Jülich and the Institute of Katrin Amundsen in Jülich, the Jülich Supercomputing Center. Many people contribute to this, and thanks a lot. And then we are at the end. We are approximately on time.
Timo, there's one more question in the Q&A.
Oh yes, one more question. Is there a difference between the MNI27 and BigBrain template aside from subjects and data available? Yes, yes, there is. So the MNI27, that is not the typical name. We typically say the Colin 27. That is a single subject average. So when you look at the Colin space, I might just do that here. If we go to the Colin space, you will see that it really shows the brain morphology at some detail, because it's a single subject average. So you can look here deep into the sulci. And if you select the ICBM 152 template, which are both from the MNI templates, you see that this is a multi-subject average. So it has a very different quality. This one is used much more in the community, and you see that this is much more smoothing. It doesn't have so sharp individual characteristics, but then it represents for sure the population much better. So these are very different. They are both at the millimeter scale based on MRI imaging, and the BigBrain has the resolution of 20 micrometer. So it's very different. It is based on one particular subject only, and has much greater detail and resolution. So it cannot be compared. Yeah, it's one individual subject, but there you have the really much more detail. Yeah, we have looked at that. So you see, you can zoom in and see the individual, at least the larger ones, individual cells. You can see the cortical layers. This is very different, but you always have to be aware that this is a particular postmortem subject, not an average of many. So it's not as representative for the population as the other one. Alan, you're raising hands.
It's just worth pointing out that the BigBrain dataset has been mapped into the MNI 152 space some time ago. So you're operating in the same space essentially with the BigBrain.
Exactly. And there will be, did Casey already have a talk? So there's this BigBrain warp, library coming up that gives you easy access to transforming data from one space to the next.